Hello, welcome back. The topic for today's microbiology class is bacterial genetics. The genetic material of a typical bacterium, for example E. coli, consists of a single circular DNA molecule with a molecular weight of about 2 is to 10 raised to power 9 and is composed of approximately 5 into 10 raised to power 6 base pairs. Bacteria are haploid, that means they have only a single chromosome, therefore a single copy of gene. In haploid cells, any gene that has mutated and therefore is not expressed results in a cell that has lost that trait. What is mutation? A mutation is a change in the base sequence of DNA that usually results in insertion of a different amino acid into a protein and the appearance of an altered phenotype. Mutations results from three types of molecular changes. First is base substitution, that occurs when one base is inserted in place of another base. It takes place at the time of DNA replication, either because of DNA polymerase enzyme makes an error or because a mutagen alters the hydrogen bonding of the base being used as a template in such a manner that the wrong base is inserted. When the base substitution results in a codon that simply causes a different amino acid to be inserted, the mutation is said to be missense mutation. And when the base substitution generates a termination codon that stops protein synthesis, the mutation is said to be nonsense mutation. Nonsense mutations almost always destroy this protein function. Second type of mutation is frame shift mutation. That occurs when one or more base pairs are added or deleted, which shifts the reading frame on the ribosomes and results in incorporation of wrong amino acids downstream from the mutation and the production of an inactive protein. Look at this picture in which this amino acid sequence is being altered that shifts the whole frame of the reading chromosome. The third type of mutation occurs when transposons or insertion sequences are integrated into the DNA. These newly inserted pieces of DNA cause changes in the gene into which they insert and in the adjacent genes also. The transfer of genetic information from one bacterial cell to another can occur by the three methods that are conjugation, transduction, and transformation. So what is conjugation? Conjugation is the process by which DNA, either plasmid or chromosomal, is transferred directly from one bacterium to another. For the conjugation process to occur, the donor bacterium should have fertility plasmid or F plasmid that encodes proteins that mediate this process, the most important of which proteins are that form the sec pilus. The DNA transferred by conjugation to recipient bacterium is a new copy that allows the donor to keep a copy of the DNA. Plasmids carrying antibiotic resistance genes are commonly transferred by this process of conjugation. This picture shows the process of conjugation in which this donor bacterium have this F plasmid that mediates the synthesis of various proteins forming this sec pilus, and by the help of this sec pilus that is attached to the recipient bacterium, and then this F plasmid mediates the synthesis of various proteins that are involved in this process of conjugation that transfer the pieces of bacterial DNA from the donor cell to the recipient cell, and then this new recipient cell or bacterium have this F plasmid and then sec pilus transferred from the donor bacterium. Second process is transduction, that is by which the DNA, either plasmid or chromosomal, is transferred from one bacterium to another by a virus. the transferred DNA integrates into the chromosomal DNA of the recipient, and a new protein such as exotoxin are made by a process called lysogenic conversion. In the process of transduction, a bacteriophage infects a bacterial cell, and the phage enters into this bacterial cell, and then this phage replicates inside the bacterial cell, and the bacterial chromosomes are broken down into pieces, and then these bacterial chromosomes and phage reassemble and then form a new bacteriophage. That new bacteriophage infects another or new bacterial cell, and then the integrates into the host bacterial chromosome, and then this new bacterial cell acquire a new trait by the help of this bacteriophage. Third process is transformation. Transformation is the process by which DNA itself, either DNA are released from dying cells or DNA purified in the laboratory, enters a recipient bacteria. Medically, this appears to be less important than conjugation and transduction. This figure shows the process of transformation in which the bacterial chromosomes released from one bacterial cell are reuptake by the second bacterial cell, and this DNA material from one bacteria is transferred or integrated into another bacterial cell.